Hello everyone and welcome to today's lecture. Okay, so today's lecture this is of zoology. As you know, NEET is around the corner just within a month, so we really need to ace up our preparation. So today what we are going to discuss? Today we are going to discuss biotechnology. As you know, biotechnology has a really high weightage in NEET and it has been an absolute favorite topic of NEET for so many years. So, biotechnology and a very good command and understanding of biotechnology becomes extremely crucial to ace this NEET exam. As this year, you could also see that there was a significant section which was asked from this biotechnology portion. So, in this session, we are going to have discussions on a few questions of biotechnology which will help you to understand this chapter better and which will help you to navigate these questions when they are asked in the NEET examination. So, without wasting further time, let us dive into the questions related to this biotechnology portion. So, as you can see here on your screen, this is your first question, and this question goes as the DNA molecule into which a gene of interest is inserted for cloning is called, you know this answer is going to be vector, right? The DNA molecule in which a gene of interest is inserted for cloning is called vector, and you know gene of interest plus vector, this is called as your recombinant DNA. Okay, now let us move on to the next question. The next question is, cutting of DNA at specific locations became possible with the discovery of, after the discovery of, so it is restriction enzymes. Okay, as you know, restriction enzymes are also called as molecular scissors. Okay, they are called as molecular scissors, and restriction enzyme is nothing but just another name of restriction endonuclease. Okay, these enzymes are your endonucleases, and what do endonucleases do? They cut the DNA at its backbone. Okay, they break the phosphodiester bond, which helps in the cutting of DNA. And why are we saying they cut the DNA at specific locations only? Because you know restriction enzymes will function by recognizing a palindromic sequence. They will recognize their palindromic sequence and then they will cut the DNA. Next question. Which technique made it possible to genetically engineer living organisms? This is your recombinant DNA technology. It is not X-ray diffraction, heavier isotope labeling, or natural hybridization. It is recombinant DNA technology. Why is it recombinant DNA technology? Because with the help of recombinant DNA technology, you can actually manipulate the DNA. Okay, this technology allows you to manipulate DNA and RNA of living organism, and hence you can genetically engineer them. Next question. Which of the following is commonly used in gene cloning? Ribosome, lysosome, lomosome, or plasmid? So, the right answer is going to be plasmid. Why is the right answer plasmid? Because plasmid, as you know, is a very commonly used vector. In just our previous question, we were discussing that gene of interest plus vector, right? We discussed this in our previous question. Gene of interest plus vector makes your recombinant DNA, and a very commonly used vector is plasmid. Okay, so plasmid is used as a vector and therefore it helps in the process of gene cloning. When an animal is made superior by changing its genotype through introduction of foreign genes, it is called as immunization, tissue culture, artificial selection, or genetic engineering. As we discussed it, it will be called as genetic engineering. Okay, and these type of animals or plants which are made superior are called as transgenic animals. Okay, they will be either called as transgenic animals, and if it is a plant, then it will be called as transgenic plant. Now, which one is incorrectly matched? So here you have to find the option which is incorrect. So let us dive into our options. Somatic hybridization, fusion of two diverse cells, micropropagation, in vitro production of plants, callous unorganized mass of cells, and vector DNA site of recombinant RNA synthesis. As you can see, option A, B, and C are absolutely correct. Vector DNA is not the site of recombinant RNA or RNA synthesis. So your answer is going to be vector DNA. Okay, why? Because vector DNA is actually used to transfer your gene of interest into your host organism. So A, B, and C are correct. D is incorrect. Hence, your answer is going to be D. Now, the sequence recognized by EcoR1. The sequence is very important because this has time and again been asked in NEET. Okay, so first of all, this sequence is a palindromic sequence and it is a sequence of 6 base pairs. So it is palindromic sequence and it is a sequence of 6 base pairs. Okay, so we have 6 base pairs here. And also you have to remember that EcoR1 always produces sticky ends. Okay, it always produces sticky ends. It does not produce blunt ends. Now coming here, what is the sequence of EcoR1? So you should remember it. It is just a memory based question. It is going to be GAATTC. Okay. So as you can see in the given options, option A, GAATTC, and we do not even need to look at option B, C, and D because we know GAATTC and GAATTC. Hence, our option A is going to be correct. You should remember this sequence because it is well mentioned in NCRT and it has also been asked in previous NEET exams. Okay. So GAATTC is the recognition sequence of EcoR1. Now, let us look at the next question. A foreign DNA and plasmid are cut with the same restriction enzyme before joining. Yes, it is very crucial that you cut your foreign DNA or gene of interest and plasmid, which is your vector, with the same restriction enzyme. Why? Because if you cut both of them with the same restriction enzyme, they are going to produce sticky ends. Okay, they are going to produce sticky ends. These sticky ends are going to join together. These sticky ends will form hydrogen bond naturally, and your process will be much simpler and easier. So let us look at our options and let us try to understand which option actually justifies this. To destroy the plasmid, no. To produce compatible sticky ends, yes. Remove selectable markers, no. To stop replication, no. So our correct answer is going to be to produce compatible sticky ends. Okay. Now let us look at our next question. Which enzyme joins DNA fragments by forming phosphodiester bonds? Okay. This enzyme is also called as molecular glue. You can very well understand why it is molecular glue because it is doing the work of joining two separate DNA fragments. And which enzyme is this? It is your DNA ligase. Hence, your option A is going to be correct. It is going to be your DNA ligase. Okay, this one is your DNA ligase. And always remember, DNA ligase belongs to the sixth class of enzyme. There are total 6 class of enzymes, and it belongs to your last class. It belongs to ligases, and it belongs to the sixth class of enzymes. Now let us look at our next question. The sequence that controls replication and copy number of linked DNA in a vector is okay. The sequence is very important, students, because if your vector does not have the sequence, the vector cannot be used in the process of genetic engineering. For anything to be a vector, one of the most crucial thing is to have a sequence which will control replication and copy number. And what is that sequence? Selectable marker, no. Recognition site, no. Prop site, no. It is going to be your ORI site. ORI is, if we expand ORI, it is basically your origin of replication. Okay. So it is the site in vector from where replication begins. And you know, when you introduce your foreign gene into vector DNA, you want your foreign DNA to replicate. So for that, your vector should have an ORI site. Okay. Now, a selectable marker is used too. This is very important. Why do we use a selectable marker? You know, when we study the important components of a vector, one of the components is ORI site that we discussed previously. And one more important thing to have in a vector is to have a selectable marker. There are two selectable markers that you study. One is antibiotics, okay, gene coding for antibiotic resistance, and the another one is LAG-Z gene. But what do we use this selectable marker for? To cut DNA into fragments? No. Produce insulin? No. Denature DNA? No. It is to identify transformed cells because recombinant transformants actually help you to achieve your goal and to get your product from genetic engineering. Okay, so a selectable marker is used to identify transformed cells. Now let us look at next question. Which of the following is not a typical feature of plasmids. Okay, so plasmids are very commonly used vector, and plasmids have some qualities. What are those qualities? First of all, your plasmids are always found in bacteria. Then they are extra chromosomal DNA. Okay, they are extra chromosomal DNA, and also they are single and double stranded. They are double stranded as well as circular. So these are the four qualities which are present in plasmid. Now let us look at the option and try to understand which of these will not be a feature of plasmid. So circular structure, yes, your plasmid is circular. Independent replication, this is correct. It is extra chromosomal and it replicates independently inside a bacterial cell. Double stranded DNA or single stranded structure. Okay. So double stranded DNA होता है आपका plasmid. Your plasmid is not single stranded structure. So single stranded structure will not be a feature of your plasmid. Now let us look at next question. Which bacterium is commonly used to transfer foreign DNA into crop plants? This is purely a memory based question. You should always remember typical vectors. For example, in plants, the common vector that you use is TI plasmid. Okay, and TI plasmid, from where is your TI plasmid obtained? It is obtained from Agrobacterium tumefaciens. So option B, your option B, Agrobacterium tumefaciens is going to be correct. Rest all other options are going to be wrong because the source of TI plasmid is Agrobacterium tumefaciens. Now, the TI plasmid is modified in biotechnology mainly too. Okay, now this question actually tests your knowledge regarding disarmed vectors. There is a concept that you study, and you study this concept of disarmed vector for GI plasmid as well as retroviruses. Okay. Now, this GI plasmid was normally a tumor-inducing plasmid, but it has been dismodified. It has been, disarmed or modified to do what? To transfer desired genes into plants. Hence, your option B, to transfer desired gene into plants, is going to be correct. Now, let us look at next question. Which vector is commonly used for introducing DNA into human lymphocytes? Okay, introducing DNA into human lymphocytes. This is a typical case which you study for ADA deficiency. So, when you study this case of ADA deficiency in biotechnology and its application, you find that there is a vector which is used to transfer your modified or good lymphocytes into the body of your patient. And what is that vector? That vector is your retrovirus. Okay, that vector is going to be your retrovirus. So this is your option A, retrovirus. Retrovirus is used to transfer the desired gene into animal cell. TI plasmid is used for plant cells. Okay, TBR 322, which is your modified plasmid, is generally used for bacterial cell, and myeloid ogine incognitia, it is not a vector, it is actually a parasite of tobacco plants. Okay, so your option A, which is retrovirus, is going to be your correct option. Now let us look at next question. DNA fragments move towards the anode during electrophoresis because DNA is. Now, here we are talking about gel electrophoresis. We are talking about agarose gel electrophoresis, where DNA are loaded into DNA wells, and under the influence of an electric field, they migrate towards the positive terminal. Why do they migrate towards the positive terminal? Because your DNA is negatively charged. Okay, they are negatively charged. Hence, under the influence of an electric field, they move towards the positive terminal. DNA is negatively charged due to the presence of basic amino acid and phosphorus. Okay, positively charged no, neutral no, hydrophobic no. Okay, your answer is going to be negatively charged. Now, again, agarose gel electrophoresis. In agarose gel electrophoresis, smaller DNA fragments move farther because they. Okay, so you have to remember one concept. In agarose gel electrophoresis, DNAs are not separated. They are not separated according to the charge. Why? Because all DNA molecules are going to be negatively charged. Hence, all of them are going to move towards the positive terminal. Then, on what basis are your DNA fragments separated? They are separated on the basis of size. Basically, you know that your agarose gel produces a sieving effect. Okay, under this sieving effect, the smaller DNA fragments can move faster, whereas the larger DNA fragments will be slow to move. Okay, so let us look at our options. The smaller DNA fragments move further because they are heavier, no, are positively charged, no, bind more strongly to agarose, no, pass more easily through gel pores. Yes, your agarose gel produces a sieving effect, and under that sieving effect, smaller DNA can pass easily, whereas larger fragments are going to take significant amount of time to pass, and their passing through the agarose gel is also not going to be easy. Now let us look at next question. Separated DNA fragments in agarose gel are commonly visualized using. Yes, as you know, DNA can never be seen with naked eye. Okay, so once you have completed your agarose gel electrophoresis, how are you going to visualize your DNA? So DNA can be visualized after staining it with stadium bromide, but this one step in itself is not enough. Okay, after staining it with ethidium bromide, you have to follow it with the exposure of UV light. So you stain the DNA with ethidium bromide, then you give it UV light, and when you do these two things, your DNA will appear as bright orange colored bands. Okay, so let us look at our options. A, no. B, yes. C, no. And D, no. Okay, so your option B, which is ethidium bromide under UV light, is going to be correct. Now, there is one more question. A 500 base pair and a 2000 base pair DNA fragment are loaded in the same agarose shell, which will move farther. Now, if you look at this question, student, this question is actually an application based question. You already know the theory behind this. We have discussed the theory behind this. We here in this question, we just need to apply that theory. Now, a 500 base pair and a 200 base pair DNA. That means 500 base pair DNA is going to be smaller, whereas your 200 base pair DNA is going to be larger. Okay, so you have one smaller fragment and then you have one larger fragment. They are loaded in the same agarose gel, which will move farther. As you know, the smaller DNA fragment, because it receives less resistance from the agarose gel and it can move easily through the gel piece. It will cover more distance as compared to a 2000 base pair DNA fragment. Why? Because this 2000 base pair DNA fragment is going to be a large DNA fragment. So which will move farther? Which will move farther means which DNA fragment is going to cover more distance. So the answer is going to be 500 base pair fragment. Okay, 2000 base pair fragment is going to cover less distance than 500 base pair fragment. Equally, no, they are not going to move equally, and neither will move. This is wrong because DNA is a negatively charged molecule, and under the influence of electric field, they will always move towards the positive terminal. Now, the correct order of PCR steps is, this is a very important question. As you know, in this year NEET paper also this question was asked. The correct order of PCR steps. So you know PCR, in the PCR the first step is already always denaturation. Now denaturation is followed by annealing, where you apply your primers, and then this annealing is followed by extension. Now let us look at our option. Which option justifies this? So option B. Okay, option B, denaturation, annealing, and extension. This demonstrates the correct order of steps in your PCR. Now, Taq polymerase is used in PCR because it is okay. So in PCR, we discussed that the first step is denaturation. And in denaturation, you provide very high temperatures, basically more than 90 degrees Celsius. Now in denaturation, you need an enzyme which can actually tolerate such amount of high temperature without getting disintegrated. So Taq polymerase is used in PCR because it is cold sensitive, no. A restriction enzyme, no. A ligase, no. But because it is heat stable, okay? Because in PCR you have the step of denaturation where you provide very high temperatures. So you require an enzyme which can sustain this high temperature without getting disintegrated or without becoming inactive. Okay, so Taq polymerase is heat stable. It is hence used in PCR, and Taq polymerase is obtained from Thermus aquaticus. Okay, so your Taq polymerase enzyme is obtained from Thermus aquaticus. This Thermus aquaticus, you know, this is a bacteria which is found in your hot springs. Okay, it is a natural bacteria. It is found in hot springs. Taq polymerase occurs naturally in it, and it can tolerate high amount of temperature. Now, in PCR, heating to about 94 to 95 degrees Celsius is done to. Yes, here basically you are being asked what happens in denaturation. When you do denaturation of DNA, what happens in denaturation? Join DNA fragments, purify DNA, bind primers. No, in denaturation, you separate the two DNA strands, right? So, as you know, in PCR, there are basically three steps. So, in denaturation, which occurs at, you have to remember the temperatures, they are important, which occurs at approximately 94 or more than 90 degrees Celsius, you separate the two DNA strands, right? In denaturation, there occurs separation of DNA strands. Now, the next step, which occurs in PCR, is your second step, which is annealing. Annealing occurs at an approximate temperature of 54 degrees Celsius. And what happens in annealing? In annealing, primers are attached. Okay, so basically, you attach primers, and this is called as annealing. The third step is extension. Okay, extension which happens at 72 degrees Celsius. And what do you do at extension? In extension, basically, you Taq polymerase, which is a type of DNA polymerase found in Thermus aquaticus, extends the DNA fragments. Okay, it extends the DNA fragments. So these are the three basic steps of PCR. Okay, so in heating to about 94 to 95 degrees Celsius for denaturation to separate the two DNA strands. Now let us look at our next question. PCR is useful in early diagnosis because okay, now this is one application of PCR that you study, and this application you study in molecular diagnosis. When we study about diagnosis, we know that there are conventional methods for diagnosis, such as there is urine culture. Okay, you can test urine, you can test blood, but those conventional methods, okay, those are the conventional methods of diagnosis, and they are usually not preferred. Why? Because those conventional method of diagnosis, they take their sweet time, they take their ample amount of time to diagnose a disease for you. But if you want a disease to be detected at very early stage, then what are you going to prefer? You are going to prefer PCR. Now, why are you going to prefer PCR? Because PCR has a very, very good application. Now, what is that application of PCR? PCR can amplify, basically PCR, what is the job of PCR? It is to amplify the DNA. So your PCR can amplify small amounts of DNA. Now what is the meaning of this amplification of small amounts of DNA? So your PCR can synthesize a very huge quantity of DNA from a very, very less DNA sample. Okay. So it can amplify small amounts of DNA, which can actually help you to detect those DNA and help you to detect an early diagnosis of the disease that you might be suspecting. Okay. Due to this reason, PCR is also useful in DNA fingerprinting. So PCR is useful because it amplifies small amounts of DNA. Now, during DNA isolation, chilled ethanol is added to. Yes, during DNA isolation, you use various enzymes, and at the end step, you add chilled ethanol. What is the job of this chilled ethanol? This chilled ethanol is actually used to precipitate DNA. Okay, so your chilled ethanol is used to precipitate DNA, which is your option B. And after precipitation of DNA, you know the DNA coils around a rod, which is called as, and this DNA can be separated out via a process of spooling. Okay, so to actually extract or isolate your DNA from the cell, you add chilled ethanol after the application or addition of all the enzymes. When you add chilled ethanol, the DNA gets precipitated, and then this precipitated DNA can be removed out through the process of spooling. Now, which enzyme is used to digest the fungal cell wall during DNA isolation? If you have fungal cell wall, you know that the cell wall of fungus is made with chitin. Okay, chitin is a substance which is also found in your arthropods. Okay, the exoskeleton of arthropods is also made up of chitin. The cell wall of fungus is also made up of chitin. So to dissolve chitin, which enzyme are you going to use? Lysozyme, no. Cellulase, no. Amylase, no. Your answer is going to be chitinase. Okay, so you are going to use chitinase enzyme to digest the fungal cell wall. Now, next question, which pair is incorrectly matched? Bacteria, lysozyme. This is correct. Why? Because to dissolve bacterial cell wall, you always require lysozyme enzyme. As you remember, this lysozyme enzyme is also present in our tears, and it is also present in our saliva. Why? Because it has antibacterial properties. What is the meaning of this antibacterial properties? It can destroy the cell wall of bacteria. Okay, so to dissolve a bacterial cell wall, you will always have lysozyme. Plant cells, cellulase. Again, correct. Why? Because to dissolve plant cells, you require cellulase enzyme. Why? Because plant cell wall is made up of cellulose. Okay. For bacteria, you will have lysozyme. For plant cell, you will have cellulase. For fungi, chitinase. Okay, you have studied for fungus, you need chitinase. We just discussed it in our previous question. So option A, B, and C are correct. D is incorrect. Okay, for algae, you will not be using methylase. Now, next question. A DNA or RNA segment tagged with a radioactive molecule is called again, a very important step that you use in molecular diagnosis. Okay, you use this in molecular diagnosis. You do this when you want to detect a disease at a very early stage. Why do you need to detect a disease at a very early stage? Because the earlier you detect disease, the quicker or the more effective its treatment plan begins. Okay, so a DNA or RNA segment tagged with a radioactive molecule is called as a probe. Okay, it is not called vector, not plasmid, not clone. It is called as probe. So probe is a small segment. It is basically an oligonucleotide which is tagged with a radioactive molecule. And then again, with the help of autoradiography, you can find the gene which is mutated, and this will help you in earlier diagnosis of a disease. Next question. Which equipment is required for large scale production of enzyme using microbes? Okay, so this is basically large scale production. The key word here is large scale production. Now in biotechnology, whenever we talk about large scale production, the large scale production of enzymes or proteins or any recombinant DNA product is done using bioreactors. Okay, bioreactors are large vessels. Basically they have a capacity of 100 to 1000 liters, and these are the vessels where you grow your host cell in a very appropriate environment to give you the maximum product. Okay, so the answer is going to be bioreactor. Next question, stirred tank bioreactors. Now we are talking about bioreactor. You know that bioreactors are of two types. One is Pager, sparged bioreactor, and the another one is stirred bioreactor. Now stirred tank bioreactors, they are called so because they have a stirrer. Okay, this stirrer, what is this stirrer? Basically, if you have water in a glass and you put salt in that water, and you will use a spoon. Okay, you will use a spoon. So this spoon will work as stirrer. Why? Because it will help you to distribute or to dissolve the salt very quickly and very easily in water. So this stirrer, what does this stirrer does in bioreactor? This stirrer actually ensures that the nutrients and the oxygens are distributed evenly and uniformly throughout the bioreactor. Okay, so stirred tank bioreactors are mainly designed to ensure proper mixing and oxygen supply, and proper mixing and oxygen supply is done with the help of a stirrer. Moving on to our next question. The separation and purification of expressed protein before marketing is called as, this is called as downstream processing. Okay. Next question, which of the following is not a component of downstream processing? Separation, yes, you separate the product in downstream processing. Purification, yes. Preservation, yes. Gene expression is not a part of downstream processing. Gene expression is actually a part of upstream processing. Gene expression is something that you do before downstream processing. Now, BT cotton is resistant to. Okay, so BT cotton here, this BT actually stands for Bacillus thuringiensis, and this BT cotton is a genetically modified product. And this BT cotton was created because cotton plants were getting very much affected by cotton bollworms. Okay, so this BT cotton is resistant to your insect pest. Okay, insect pest, which was your cotton bollworm, which was actually destroying a large quantity of your cotton plant. Now, Bt protoxin becomes active in the insect gut due to. Okay, so as you know, Bt crystals, they are inactive protoxins. Okay, they do not harm the bacteria in which they are produced. Why do they not harm the bacteria in which they are produced? Because in bacteria they are present as inactive protoxins, but they can kill insects. Why can they kill insects? Because in insects they get activated. Okay, they get activated in insects. And why do they get activated in insects? The answer is alkaline pH of the midgut. Okay, so in the insect midgut, there is an alkaline pH, and this inactive Bt protoxin gets activated in the alkaline pH of the insect. Now, the activated Bt toxin kills insect by, yes. So this activated Bt toxin, how is it going to kill insect? So it actually creates spore. Okay, it creates spores in the wall of midgut epithelial cells, and due to this spore creation, the insect is going to die. So preventing oxygen transport, no. Photosynthesis, no. Digesting insect DNA, no. Your correct option is going to be option A. They are going to destroy the midgut epithelial cells. Now moving on, which statement about bt crops is correct? Okay, so let us, there are four statements here. You have been given four statements, and we have to find out which statement actually justifies bt crops. Okay, now bt toxin is coded by crygenes, this is correct. Bt toxin kills all the insects, no. Bt toxin is very insect specific, you know, crygenes are insect specific. Bt toxin activates in acidic gut, no, it activates in alkaline pH. Bt toxin is harmful to the humans in the same way. No, Bt toxin is very insect specific, so it will harmful a certain kind of insect, certain class of insects. It is not going to harm humans. So the correct option is going to be option A, which is Bt toxin is coded by cry genes. Now, golden rice is genetically modified. So as you know, children, golden rice is actually a transgenic plant. Okay, it is a transgenic plant. Now, golden rice was created artificially. Why was it created artificially? Now, why is it called as golden rice, first of all? Because it has a slightly golden color. It is slightly golden in color, slightly yellow in color. Why is it so? Because it has a gene. It has beta carotene gene. Okay, it has beta carotene gene which gives it that color. But with that, it also helps in the production of vitamin A. Okay, so golden rice is genetically modified to improve vitamin A content. Now you know this vitamin A, it is very essential. Why? Because vitamin A prevents you from night blindness. Now, RNA interference causes. Okay. Now, RNA interference, which is written as RNAi, you know, RNA interference is a natural process which takes place in all eukaryotic organisms, and it takes place as a method of natural defense. Okay. It is a defense mechanism which operates in all eukaryotic organisms. And how does RNA interference helps in defense? Increased translation of all mRNA? No. Silencing of specific mRNA, yes. Increased mutation rate, no. Direct DNA replication, no. As you know, RNA interference, as you have studied, RNA interference is a, what do we call it? We say it is a post transcriptional modification. This is what we say about RNA interference. We say that it is a post transcriptional modification. So, if it is a post transcriptional modification, what will it do? It will actually stop the translation of a specific mRNA. Okay, so your correct option is going to be option B, silencing of specific mRNA. Now, next question. In RNA interference, gene silencing is triggered by. Yes, so in basically this question, we are asking you what is that one process, what is that one starting point which will, you know, escalate or start this entire process of RNA interference. So, you know, in eukaryotic cells, the RNA is single stranded. Okay, whenever a double stranded RNA is formed, whenever a double stranded RNA is formed, the source of other RNA being for a viral genome or from a transposon. Whenever this double stranded RNA is formed, your RNA interference process gets activated. So, this RNA interference, which leads to gene silencing, is triggered by the formation of double stranded RNA. Now, let us look at next question. RNA interference technology has been used to protect tobacco plants against. Yes, you have to remember the name. It is Meloidogyne incognitia. Okay, so it has been used to protect tobacco plants against Meloidogyne incognitia, which was causing your root knot disease. It was causing root knot disease in your tobacco plants. Now, flavor saver tomato was developed to Now, this was a special kind of tomato, flavor saver tomato, and it was actually developed to increase shelf life. Okay, it was developed to increase shelf life. Okay, there was one gene which was responsible for the ripening of fruit. That gene was inactivated using RNA interference, so that flavor saver tomato can actually have a very good shelf life. Vitamin A, no. Insects, no. Protein contents, no. The correct option is going to be option B, which is going to be delay ripening and increase shelf life. Now let us look at next question. Herbicide resistant crops are developed mainly to herbicide resistant crops against they are transgenic. Okay, they are resistant to herbicides. Okay, now you know these herbicides, these pesticides, they cause a lot of pollution. Okay, herbicides and pesticides cause a lot of pollution in our environment, and these herbicides and pesticides are not good for the environment as well as for the humans in the long run. So why are these herbicide resistant crops produced? To increase vitamin content? No. Prevent viral disease in humans? No. Produce antibiotics? No. To eliminate weeds without harming crops. Okay, so the principle or the vision behind creating herbicide resistant crops, or for that matter pesticide resistant crops, pesticide resistant crop, as you know, bt cotton, it was to create a sustainable environment. Okay, so herbicide resistant crops, they can be eliminated, they can eliminate weeds without harming crops. Now, which is not a direct benefit of genetically modified crops? These GMO or genetically modified organisms, genetically modified crops are just, they can also be called as transgenic plants. Okay, so what are the benefits of transgenic plant? Do they reduce pesticide use, improve nutritional quality, automatic increase in soil fertility, and stress resistance? You know, they reduce pesticide use. This is correct. They also have improved nutritional quality. One example being golden rice, which is very rich in vitamin A. Stress resistance, yes, genetically modified crops actually tolerate drought and other natural stress better than non-transgenic crops, but they do not increase automatically, they do not increase the fertility of soil. So option C is not going to be a direct benefit of genetically modified crops. Okay, so let us look at our next question. See, in this we are getting asked about transgenic plant. A transgenic plant, which can also be called as genetically modified plant, is one that is produced only by hybridization, produced by natural pollination, regenerated after introduction of foreign DNA, and always sterile. So your genetically modified plant is regenerated after introduction of foreign DNA. Okay, what is a genetically modified organism or a transgenic plant or animal? It is a plant or animal whose DNA or RNA has been altered artificially to actually express a foreign gene. Okay, so a transgenic plant is no different. It is just, it is regenerated after introduction of foreign DNA. Option C is going to be correct answer. Now, next question. Recombinant human insulin is preferred over animal insulin because, okay, animal insulin, as you know, this was actually obtained from cattle. Okay, it was actually obtained from cattle before the development of recombinant human insulin by Eli Lilly, and it was not preferred. Actually, it was not preferred. Why? Was it less effective? No, it was effective. Causes fewer allergic reactions, has no peptide chains, cannot be mass produced. Animal insulin was actually not preferred because it could cause allergic reactions. Okay, this animal insulin was actually reported to cause allergic reactions in several patients. So that is why recombinant human insulin is preferred. Why is it preferred? Because it actually causes fewer allergic reactions. Okay, so your option B is going to be correct. It is going to have peptide chains. A recombinant human insulin can actually be mass produced. This is one of the reasons why we appreciate or why we prefer recombinant human insulin so much over animal insulin. Okay, the correct option is going to be option B because it causes fewer allergic reactions. Now let us look at next question. Recombinant insulin production involves separate production of, as you know, if you talk about a mature insulin has chain A, it has chain A and chain B. Okay, it has chain A and chain B. It does not has chain C because chain C is present in an immature insulin or inactive insulin or proinsulin. So your mature insulin or active insulin has chain A and chain B, which are joined by disulfide bonds. So it involves separate production of A and B chains. Now, which one is not a product of recombinant DNA technology? So, in this question, you have basically been given four options. Okay, these are four products, these are four things. Out of this, three things have been produced, already produced using recombinant DNA technology, and one of them has not been produced. So, let us look at our options. Human insulin, yes. Eli Lilly. Interferon, yes. Human growth hormone, yes. Penicillin, no. Penicillin is not produced using recombinant DNA technology. It is produced naturally using your natural microbes. Okay, so penicillin is not produced using recombinant DNA technology. So your answer is going to be option D, penicillin. The first clinical gene therapy was performed for. Okay, so your first clinical gene therapy was performed on a 4-year-old girl who was suffering from ADA deficiency. Okay, she was suffering from ADA deficiency. Okay, adenosine deaminase deficiency. Adenosine deaminase is an enzyme which is very, very important and very critical for the maturation of lymphocytes. So it was performed for ADA deficiency. Now let us move on towards next question. In gene therapy for ADA deficiency, the functional ADA gene is introduced into. Yes. Where do you actually introduce your functional ADA gene? In which blood cell do you do this? So the answer is going to be the functional ADA gene is introduced into patient's lymphocytes. Okay, because in the absence of ADA, immature lymphocytes are produced. Okay, so your lymphocytes are not mature. As you know, your lymphocytes form a major part of your immune system. So if you have immature lymphocytes in your blood, your immune system is going to get compromised. So this functional ADA gene, this is introduced into patient's lymphocyte, due to which your non-functional lymphocytes actually become functional lymphocytes. Now let us look at next question. Gene therapy may not give permanent cure. If yes, see, the gene therapy is very helpful. It has been very, very useful in providing relief to patients suffering from ADA deficiency, but it is not a permanent solution. Why is it not a permanent solution? Because as you know, your lymphocytes, the lymphocytes are the cells in which your ADA gene is introduced. Your lymphocytes are not immortal. Okay, your lymphocytes are not immortal. They are actually the cells which will die within a few weeks to few months' time. So, as your lymphocytes are not immortal, so the corrected lymphocytes that you have introduced, they will die within a few weeks' time. So for this, what do you need to do? You need to again and again do this process of gene therapy where you will introduce your functional ADA gene into lymphocytes. So you have to keep on doing this. It is not a permanent cure. Why? Because your corrected cells are short-lived. What are your corrected cells here? These are lymphocytes. Okay, your lymphocytes are the corrected cells here, and these lymphocytes, they are not immortal. They will die in some short period of time, and then you have to again do a process of gene therapy where you will again introduce your functional lymphocytes into the body of the patient. Okay, so the correct answer is going to be A, because your corrected cells are short lived, and these corrected cells are none other than lymphocytes. Now, ELISA technique, as you know, ELISA is enzyme linked immunosorbent assay, is based on DNA amplification, no. RNA interference, no. Protein crystallization, no. It is based on antigen antibody interaction. ELISA is basically used in molecular diagnosis. Okay. And as you know, ELISA is a very common diagnostic method which is used to detect AIDS or HIV. Okay, so ELISA is a technique which is used to detect AIDS, and ELISA, the principle of ELISA is based on antigen and antibody interaction, which is basically every antigen is specific to a particular antibody. Now, in blue white screening, colonies containing recombinant plasmids appear white because, okay, so colonies containing recombinant plasmids appear white, whereas non recombinant colonies appear blue. Why is that? Because here LAG-Z gene is deactivated. Why is it deactivated? It is deactivated due to insertional inactivation. Okay, by a process which is called as insertional inactivation, where the insertion of your foreign or desired gene is going to make the LAG-Z gene inactivated. Okay, so when your LAG-Z gene is activated, your XGAL will not be converted into a blue colored product. Hence, the colonies will appear fight. Okay, so your answer is going to be B, because the option says lag Z gene is insertionally inactivated. And what is inserted? Here your foreign DNA or your gene of interest is inserted. Now, next question. Which of the following is not an essential feature required in a cloning vector? Okay. So for cloning vector, there are few features which are like non-negotiable. If you want a cloning vector, these are the few characteristics that must be present in your cloning vector. Let us look at the characteristic. Origin of replication, ori site, very very crucial. Okay, if a vector does not have an ori site, it will not be useful in genetic engineering because it cannot replicate inside the bacterial cell. Okay, so origin of replication is a non-negotiable. Selectable marker, again very important. Why are they important? Because as we studied, they help in identification of transformed cells. Okay, so they help in identification of recombinant transformants. Okay, they help in the identification of recombinant transformants. Hence, they are very, very crucial. Unique restriction site, yes. What is the meaning of unique restriction site? It means that there must be a palindromic site or restriction site for your selected restriction enzyme. Okay, so there must be a unique restriction site for each restriction enzyme. So this is also necessary or important. Then ribosomal RNA synthesis site. Okay, this we do not require. We can do without this. We can do very well without this. So your answer is going to be option D. Ribosomal RNA synthesis site is not an essential feature of cloning vector. Rest all other features, which is origin of replication, selectable marker, unique restriction site, they are non-negotiable and they are required in a cloning vector. Now let us look at next question, which is interferons. Okay, interferons are produced using biotech, and interferons are they are proteins secreted by virus infected cells which protect non-infected cells from further viral infection. Now, which of the following is a recombinant vaccine? Okay, so here there are BCG vaccine, typhoid vaccine, oral polio vaccine, and hepatitis B vaccine. Out of all these four options, there is one vaccine which has actually been produced using recombinant DNA technology. And what is that? That is hepatitis B vaccine. It is actually, do you know what is the host cell? The host cell for hepatitis vaccine is a yeast cell. Okay. So the host cell for hepatitis B vaccine is yeast cell, and hepatitis B vaccine is a vaccine which is produced by using the technology of recombinant DNA. Okay, so hepatitis B vaccine is a recombinant vaccine produced by using yeast cell as host organism. Now, next question, let us look at next question. Transgenic animals are those which, what are your transgenic animals? So as we have discussed, whether you call it transgenic animals, whether you call it genetically modified animals, what are these? These are the organisms or animals which have their DNA manipulated to possess and express an extra gene. Okay, so transgenic animals have their DNA manipulated to possess and express an extra gene. Next question. The transgenic animal known for producing human protein enriched milk is, okay. So what is it? Is it Dolly? No. Is it Tracy? No. Is it Polly? No. Is it Rosie? Yes. So as you know, Rosie was basically a transgenic cow. Rosie was a transgenic cow, and Rosie produced a milk which was actually more nutritious. Why was it more nutritious? Because it contains a protein, human alpha lactalbumin. Okay, so it contains human alpha lactalbumin protein. And what was the quantity of this protein? It was 2.4 grams per liter. Okay, because Rosie can produce milk which contained human alpha lactalbumin protein. Therefore, the milk of Rosie was actually more nutritious than normal cow milk. Okay, so Rosie was a transgenic cow and it can produce a protein, human protein enriched milk. Okay, now let us look at our next question. The organization responsible for decisions regarding genetic engineering approval in India is, okay, ICMR? No. Indian Council of Medical Research. This has nothing to do with genetic engineering approval. WHO. Okay, WHO is your World Health Organization and it is not responsible for genetic engineering approval. GEAC and CSIR. CSIR, as you know, Central Scientific Research Institutes, they are connected with lab work and they are concerned with other things. They are not concerned with genetic engineering approval. Genetic engineering approval is actually done by Genetic Engineering Approval Committee, which is your GEAC. Okay, so genetic engineering approval in India is done by GEAC. Let us look at next question. Biopiracy refers to. Okay, you already know the meaning of this word piracy. Okay, piracy is basically unauthorized or illegal use of something where you have not taken the permission from the concerned party, the original authors or the original source, and you are using it without their consent, and you are also not giving them the sufficient amount of compensation that they should actually receive, being the primary owner of this product. Now, this is piracy. And when you add bio word in front of it, it means the piracy of biological products. Okay. So let us look at the definition. Legal patenting of one's own invention. No. If you are inventing, if you are just, patenting your own invention, it is not a piracy. Okay. Use of bioresources without proper authorization or compensation. This will be biopiracy. Normal tissue culture, no. Recombinant insulin production, no. So what is biopiracy? Biopiracy is actually the use of bioresources proper authorization or without proper authorization or compensation. Okay. So this was all for today. This was all for your biology zoology class. Okay. So where here we have discussed our biotech portion. Okay. In our next master classes, in our next classes, we will be discussing further questions, further topics, which will be very crucial and very important for your NEET preparation. Okay. So till then, keep on studying and don't lose hope. Stay motivated. Okay. Thank you, everyone.